Spiders have spun silk for millions of years, creating webs of beauty and lethal efficiency. But what happens when that ancient blueprint is disturbed? The story of the drugged spiders revealed something astonishing about the brain. These experiments are famous for their stark, visual results. A web spun under the influence of caffeine is a chaotic scrawl. Another, after a dose of a sleeping drug, is a pathetic fragment. These webs are portraits of a mind in disarray. They show how chemical substances can alter ingrained behaviours. Zoologist H.M. Peters consulted a pharmacologist, Peter N. Witt, who suggested giving spiders caffeinated water. The spiders' webs became disastrously distorted. What started as an attempt to manage a filming schedule became a profound exploration of neurotoxicology. To appreciate the strangeness of drugged webs, we must first understand the marvel of a normal one. The construction is a highly stereotyped sequential process. The spider begins by creating a bridge line, a single strand of silk carried by the wind. From this, the spider lays down radial threads, the spokes of the wheel, converging at a central hub. It lays the capture spiral, the sticky silk designed to ensnare prey. The final web is a triumph of evolutionary design. It is a physical manifestation of behavioral rules encoded in the spider's DNA. We are seeing a map of the spider's brain at work. The pristine geometry of the normal web serves as the control against which all subsequent chemical chaos is measured. Peter Witt's challenge was delivering a precise dose to a small spider who wouldn't willingly take a pill or injection. They used a microsyringe to place a drug-laced sugar water droplet on the spider's mouthparts. The spiders would readily drink the sweet liquid. Researchers photographed each web, measuring radial threads, spirals, web size, and thread spacing. Would different drugs disrupt the web's regularity in characteristic ways? Spiders were kept in controlled environments to isolate the drug's effect. The spider acts as a biological plotter, its web visualizing the drug's disruption of its brain. The results of the experiments were immediate and visually spectacular. Different drugs produced distinct and repeatable patterns of web degradation. Caffeine, a common stimulant for humans, proved to be a potent toxin for the spiders. Spiders given caffeinated water produced webs that were a chaotic mess. The threads were laid down in a frantic, haphazard fashion with no discernible plan. The shape was erratic, the spirals were wildly uneven, and the entire structure looked as if the spider had been panicked and confused. The web was small, and the threads seemed to be laid with no regard for the overall geometric integrity, a perfect picture of overstimulation. In stark contrast were the effects of depressants. When spiders were given a sleeping drug, such as chloral hydrate, their behavior was the opposite of the caffeine-addled spiders. They seemed to lose all motivation to build. They would start the web, laying down a few radial threads, and perhaps one or two spirals of the capture web, but then they would simply stop. The resulting structures were pathetic, unfinished fragments, evidence of a creature that had been sedated into abandoning its most fundamental task. It was as if the drug had switched off the spider's internal drive, leaving only the ghost of a web behind. Other psychoactive substances produced their own unique signatures. LSD, the potent hallucinogen, caused the spiders to build webs that were surprisingly well ordered, even more so than their normal webs, but with a rigid, almost obsessive regularity in their geometry. It seemed to distort the spider's sense of size and planning, but not its ability to execute the basic movements. Marijuana, on the other hand, seemed to cause the spider to lose concentration. Spiders, under its influence, would build the initial framework correctly, but would appear to forget what they were doing midway through the construction of the capture spiral, leaving large, unfinished sectors in the final web. Each drug, therefore, created a unique architectural fingerprint of its toxic effect. Benzodrine, another stimulant, led to webs that were energetic but irregular. Mescaline produced webs with a distorted geometry, similar to LSD but with its own characteristic flaws. It became clear that the web was a remarkably sensitive indicator of chemical interference. The scientists could, with some accuracy, identify the class of drug a spider had been given simply by examining the structure of its web. The spider's silk, spun from its own body, was writing a detailed prescription of its own poisoning for all to see. These peculiar webs were far more than just a gallery of biological oddities. They offered profound insights into how chemical substances interact with the central nervous system to alter behaviour. Because web building is such a fixed, instinct-driven action, any deviation from the norm points to a disruption in a very specific part of the spider's neurological toolkit. For instance, the chaotic webs of caffeinated spiders suggest that stimulants interfere with motor control and the ability to execute a planned sequence. The unfinished webs from sedated spiders indicate an impact on the initiation and motivation centers of the brain. The spider's web became a map of a drug's effect. What is truly remarkable is that these effects mirror, in a simplified way, what is seen in more complex animals, including humans. Caffeine makes humans jittery and can impair fine motor skills. Sedatives reduce motivation and alertness. The spider experiments demonstrated a fundamental principle of pharmacology, that many drugs have conserved effects across vastly different species. The specific neurochemicals and receptors that these drugs target in a spider's ganglion, its version of a brain, are ancient in evolutionary terms. They're related to the same systems that govern movement, planning, and execution in the vertebrate brain. The experiments provided a powerful model for toxicology screening. The web provides a clear, quantitative, and permanent record of an animal's behavioral output. One can easily measure the web's dimensions, the angles of the threads, and the size of the gaps. This allows for an objective assessment of a chemical's toxicity. A substance that causes a significant and repeatable distortion in the web is clearly neurotoxic. This principle suggests that simple organisms with stereotyped behaviors can be used as effective, low-cost screening tools to test the potential neurological impact of new chemicals before they are ever considered for use in more complex animals or humans. Ultimately, the drugged spider webs force us to contemplate the very nature of the mind. They show that complex behaviors, which we might romantically describe as instinct, are rooted in precise biological and chemical machinery. Change the chemistry, and you change the behavior. The spider's perfect web is the product of a healthy, balanced nervous system. The distorted web is the product of that same system thrown into chemical disarray. It is a stark reminder that the mind, whether it belongs to a spider or a human, is not an ethereal entity but a physical process, one that is profoundly vulnerable to the molecules we introduce into it. Decades after Witt's initial discoveries, the spider drug experiments found a new life in a rather unexpected place. The laboratories of NASA. In 1995, scientists at the Marshall Space Flight Center revived the research, but with a 21st century twist. Their goal was to develop a more precise and objective way to measure the changes in the webs. While the original photographs were visually compelling, comparing them could be subjective. NASA's team brought the power of computation to the problem, seeking to quantify the toxicity of various substances with mathematical rigor, potentially for monitoring toxins in space or on Earth. The researchers used the same basic premise: give European garden spiders controlled doses of different chemicals, caffeine, amphetamine, marijuana, and chloral hydrate, and then photograph the resulting webs. However, instead of just looking at the pictures, they scanned them into a computer. They then developed sophisticated algorithms to analyze the web's geometry. The software could measure dozens of parameters with incredible precision, things like the standard deviation of spiral distances, the variance in radial thread angles, and the number of completed cells versus incomplete ones. This turned a qualitative observation into a quantitative statistical analysis. The computer analysis confirmed and refined Witt's original findings. The algorithms could clearly distinguish between the patterns of toxicity produced by the different drugs. More importantly, they could detect subtle deviations that might be missed by the human eye. The system was so sensitive that it could measure the degree of toxicity on a continuous scale. The data showed a clear dose-dependent relationship. The more caffeine a spider was given, the more chaotic and irregular its web became, a fact the computer could express with a precise numerical value. The system was becoming a highly sensitive bioassay for neurotoxins. NASA's work demonstrated the power of combining classic biological observation with modern computational analysis. The spider, with its innate programming, provided the raw data in the form of its web. The computer provided the tools for a deep, unbiased and statistically robust interpretation of that data. This marriage of biology and technology elevated the research from a fascinating curiosity to a potentially powerful scientific tool. It showed how we could use one of the oldest behaviours in the animal kingdom as a state-of-the-art sensor, a living monitor for chemical contaminants in our environment. The story of the drugged spiders endures not just for its scientific merit, but for its powerful symbolism. The images are arresting because they make an invisible process, the effect of a drug on the brain, powerfully visible. We cannot see the chemical reactions occurring in the synapses of a human brain, but we can see their analogue written in silk. The web becomes a metaphor for the mind itself, an intricate, well-ordered structure that can be easily tangled and broken. The distorted webs serve as a potent, almost artistic warning about the fragility of our own mental architecture and the profound impact that chemicals can have on it. Of course, these experiments are not without their limitations. A spider's nervous system is vastly simpler than a human's. It lacks the complex cognitive and emotional centers that define the human experience of these drugs. The spider's web is a readout of its motor control and its ability to follow a sequential plan, not its mood or its thoughts. Therefore, we must be cautious about drawing direct parallels. The experiments tell us a great deal about neurotoxicity and the disruption of instinctual behaviour, but they cannot tell us what it feels like to be a spider on LSD. The research offers a model, not a direct translation. Despite these limitations, the fundamental lesson from the spider's web is profound. It demonstrates that behaviour is not magic, it is biology. It is the output of a physical system, and when that system is altered, the output changes in predictable ways. The experiments strip away the complexity and mystique of psychoactive drugs and reveal their raw mechanical impact on a nervous system. They show us that the same substances that can inspire art or fuel addiction in humans can also reduce the elegant geometry of a spider's web to a meaningless tangle of silk. This universality is a humbling and important piece of knowledge. In the end, the small spider, diligently spinning its world, has taught us a great deal. It has provided a window into the intricate workings of the brain and a tool for understanding how it can be disrupted. The experiments serve as a timeless illustration of the delicate interplay between chemistry and behaviour, a principle that applies to all creatures that think, act and build. The web, in all its perfection and all its potential for distortion, remains one of nature's most eloquent storytellers, a silken chronicle of the mind at work and the mind in chaos.